Well, thanks for coming. It's a great honor to be here and give the Vaclav Sapolsky lecture, and even more to give it about my favorite geneticist, perhaps, McClintock, who of course worked here for 50 years, and I will give you something of a scientific history of the discovery of transposable elements and what it means for the modern sort of history of genomics and epigenetics. But then Mila asked me to tell some more recent scientific work from, mostly from my own lab, to sort of illustrate how her ideas still have tremendous influence on everything that we do. I want to start just with this, this slide that draws attention to an article that Paul Comet and I wrote for Cell a couple of years ago now on the 25th anniversary of her death. Paul Comet, I just want to acknowledge him very importantly for this talk. He was a graduate student here in the 1980s and worked closely with Barbara, who was on his thesis committee on transposable elements. And after she died, Paul and I were asked to look through her column and her pedigrees and her genetic notes and manuscripts and all of that, which took about a year. And much of what I'm going to tell you in the first half of the talk was really sort of gained from that experience with Paul, and we wrote about it in this article. But I'm just going to leave a little bit back. Before she joined it in 1928, it was led by Rollins Emerson, who was one of the great founders of maze genetics, and had some very famous characters in the group. The joker in the group, you can easily tell, I hope, if you're in the front row, was Marcus Rhodes, who is a very famous maze geneticist, and he's holding a magnet, attempting to retain the youngest member of the group, Xi Wen Li, who actually left the group that year. He went on to go back to pre-revolutionary China, where he became one of the most famous rice breeders and sorghum breeders ever, and actually founded the Academica Sinica in Taiwan, which is, I gave a lecture in his honor a couple of months ago in Taiwan. But the next year, the group looked very different. First of all, it was smaller. You'll notice, though, that the dog is the same, George Beagle's dog. And the group, of course, included Barbara, 27 years old there on the right. And both George Beagle, of course, and Barbara McClintock went on to win the Nobel Prize. There aren't that many labs that can claim a 50% hit rate with respect to that. But Marcus was still a joker and pointed out to people later on that even the dog was smart. Okay, so Barbara actually had a rather good year. This picture was taken in the summertime during pollinations in 1929, and that year she had her first single author science paper. For those of you who thought that Barbara never published anything, when she was 27 years old, because she discovered a staining technique. She had modified a staining technique that was able to very beautifully, and I'll show you some pictures in a minute, stain maize chromosomes in a way that you could really map the location of breaks and other sorts of recombination events and so on the chromosomes. And she used this technique in 1929 to first describe all the 10 chromosomes of maize. We have this reprint. It was from her collection. It's in the library. And you probably can't see from here, but she's drawn a little arrow between chromosomes 2 and 4. We don't call them 2 and 4. She actually numbered them backwards to begin with, because she'd been able to assign genes to these chromosomes by looking at trisomics and monosomics and the phenotypes of those anticodes. And she'd actually got 2 of them the wrong way around. And later on, after she published this paper, she actually drew a little arrow to indicate the fact that they should be swapped, which I thought was quite cool. We have that reprint. But anyway, she went on to study a very interesting phenomenon known as the chromosome and chromatid breakage fusion bridge cycle. We have a very fancy modern name for this now. It's called chromothripsis. But in those days, it was the breakage fusion bridge cycle. And she got the material originally from Louis Stadler, and there was basically a rearrangement in the chromosomes that induced breaks, but random breaks, which she could map using color genes on chromosome 9, and therefore got these beautiful ears that have all sorts of spots on them according to which genes are missing because of the breaks. And actually, I took this picture at the Nobel Museum in Stockholm. We lent these ears from her collection to the Nobel Museum, which is where they are right now. And Paul noticed while we were going through this corn, and you probably can't see it in this photograph very well, but there's a single kernel missing. And the way she discovered transposable elements was because she recognized a single kernel on an ear like this, and Paul and I would love to believe that this is actually the ear, that had a different pattern of spots. And she recognized that that pattern of spots meant that there was a programmed break heading at a very specific place on the chromosome that always generated the same break rather than these random chromathripsis-like rearrangements. And she called that locus dissociation, because it dissociated the chromosome. And that is DS, and that turned out to be the first transposable element. She used her staining technique to very accurately map that break. And if you're sitting in the front row, you might be able to tell this, but it's pretty amazing that she was able to do this. The break is illustrated here, C, on this camera lucida drawing, but if you look at the stained chromosome, it's quite remarkable that you can really see that break. And she was able to, I realized by drawing these out and measuring them that they always occurred at the same time. But importantly, she could use genetics to be able to map the break with respect to other genes on chromosome 9, which happened to be her favorite chromosome, and she'd been mapping genes there for a very long time. And I'm just going to sort of give you a very layman's version of May's genetics to help you get through this. So she used a number of genes on chromosome 9 of May's that control the color of corn kernels. So you're probably used to eating yellow or white corn, but naturally corn is actually purple, and it's because of anthocyanins, the exact same pigment that's in red wine, and in fact the same genes control that pigment in all plants, including maize. The most important, probably, we now know, is a transcription factor. It's called C1. Maize genes are named after the mutant phenotype, so C1 stands for colorless, okay? And is absolutely required for seed color. I inhibitor, we now know, the scientists in the audience, is a dominant negative form of this transcription factor. And so if you have this gene, even in one copy, the kernels are going to be colorless. Okay. Bronze encodes an enzyme that changes the color from bronze to purple. And so it's a simple color change that is also on chromosome 9, it's closely linked to these other genes, and so she was able to use this to great effect. Waxy, I'm not going to talk about very much, but it turned out to be the gene that allowed the molecular cloning of these transposable elements. Waxy is a starch by a synthetic enzyme, and she would use this beautiful iodine staining technique to be able to see waxy both in the pollen and in the seed. Finally, A1 is another anthocyanin-less gene, so it's another biosynthetic gene that controls pigment. If you have the gene, you're purple. If you don't have the gene, you're not. Okay, I hope that was clear. that's one chromosome arm of May's genetics, roughly. Okay, so how did she tell that chromosomes were breaking at a specific point on chromosome 9? Well, the order of genes on chromosome 9 is as follows. There's colorless, the dominant form, so normally give you a colored kernel. But right next to it is little bronze, which would therefore give you a bronze kernel. On the other homolog, you have the dominant negative inhibitor, so you're now white, but linked to the bronze wild-type gene, so you'd be purple if you weren't white. She could map DS because breaks at that position removed that part of the chromosome and revealed a large C, small bz, and otherwise a bronze sector. So what she saw on that famous ear, on that single kernel, were little bronze sectors all the same size, which told her that breaks were always occurring at the same point on the chromosome and must be due to a programmed break, and she called that DS, dissociation. Now, the crucial observation, of course, for her discovery was that DS could move. It could move from one location on the chromosome to another. And how did she figure that out? I'm just going to skip over these This is what I give to the Watson School students every now and then. When I give these lectures, they all get completely confused, with a few exceptions. She could tell that DS had moved because she noticed a different pattern of coloured spots on those kernels. And the way this worked, and she could also tell genetically that it needed another locus to activate it. I'll come back to that. So just to remind you, if DS is proximal to bronze, you end up with these breaks that give you bronze sectors. But if DS has moved to a location between C and BZ, now those breaks will remove I, so release color, but now those sectors will be purple. And so the reason she discovered the difference between one location of DS and the next was because the sectors were now purple. And she writes about this in her Carnegie Institute report in 1948, in case you don't believe me. And in case you don't believe me, we have the actual ear at the back of the room that has those purple spots. And Paul and I were able to find that because she carefully saved these ears and as a demonstration for this, and I'll just guide you through that. I haven't told you this yet, but she showed that there was an unlinked locus that was required to make the spots, to make the breaks, and she called that activator, and that turned out to be the first autonomous transposon that could actually activate other transposons, and that's why it's called AC. These were the ears that she'd saved. We have one of them at the back of the room. This is the one with the purple sectors that showed that DS had transposed. And she'd written, this is her handwriting, which was famously terrible, where she had all the genotypes written out on those cards. And she kept meticulous pedigrees, of course, as a geneticist, of every single plant in the field, each of which had a number. And so on the backs of these cards are the numbers that correspond to those actual crosses. And so Paul and I went back through her cards, which she'd also left, and kept. They're now at the American Philosophical Society, but we have photocopies of them all here, and I still use them. I used them this morning to make sure I got this right. And we were able to. So, for example, 5221 there is actually the parent of that cross that had the transposed DS, and we went back to that concurrent. 5221. Here's the genotype. DS is between I and sharpie. And then following back the pedigree, we could see that again in the generation before, DS is in the transposed position. And then the year before, the parent of this cross, famously 4306, and I gave this lecture recently at the Mays Genetics meeting, where I was greatly honored to be given the McClintock Prize last year. And I mentioned the number 4306. And people from Sue Wessler's lab said, yes, 4306. Yeah, big guy. You can tell by the number of different phenotypes that she'd scored, she was pretty excited about 4306. And if we go back to the generation before, where she got that ear, she'd actually written on the card, new DS locus. And that's literally the first transposition recorded of any transposon ever, because she had discovered it. And that was a very exciting finding for us, and as I said, we still have those ears. So, oops. She, of course, was working. That was all done in the summer, winter greenhouse, and following summer of 1947 and 1948. And she wrote about it in those Carnegie reports with exactly that work. She was, of course, working at the time in what was known as Animal House, since being renamed McClintock Building. And she was occupying space for visiting scientists at the time. But she was replaced in 1953 when she moved just 20 meters or so to Demarek, which was a new building in 1953, and she moved into the corner office there, and was replaced in the Animal House by Vaclav Szybalski himself. So he took her space, and I'm sure he remembers that. But her co-occupants were a pretty amazing crowd. They included Al Hershey, Kelvin Bridges at one time, Riddle, who's a very famous mouse geneticist who ended up setting up the Jackson Labs. It was a pretty outstanding group. This is just the map there, and this is where she was. She was on the second floor corner office there, and replaced by Vaclav later on. Of course, 50 years later, or 40 years later, the building was renamed McClintock Building. and there was a dedication ceremony, and I just borrowed this from the brochure that Mila kindly gave me. Animal House was renamed the McClintock building, and of course it was at that time occupied by the famous David Beach, as well as Dick Lacombe, and they were just starting the first genome project on campus, which was actually the sequence of the fish and yeast genome. And I just had to show this picture of Dick because I always knew that Dick was in Animal House. Really a remarkable likeness for those you can see John Bushy's picture. So she moved, of course, to Demerich and then continued her work there. And in fact, she worked there for the rest of her life, and that's what her column was stored. So obviously, after her discovery of transposons, it took quite a while for everyone to recognize that there were transposons everywhere. It turns out that about half the human genome is transposon. Something like 80% of the maize genome is transposons. So perhaps it was no surprise that she found them there. But it took a while for people to realize that. It turns out that there are two broad classes of transposons. We call class 1 retrotransposons. These are actually not the ones that she discovered, but they also transpose, and they have an RNA intermediate. This will become important later. But the class that she described were DNA transposons, pieces of DNA that can literally jump around the genome. These are now called class 2 DNA transposons. They were found in many different species. The first recognition that they were actually very common was the discovery of bacteria by people like, and I've put some names on these slides, and I'm sure I'm going to get criticized for them, but hopefully they won't be recorded. But Heinz Sadler and Peter Starling was actually a very good friend of ours, were some of the first bacterial geneticists to discover them and actually switch from bacterial genetics to plant genetics because they were so excited about her work, and they worked with her material to identify some of the plant transposons that she found. In bacteria and eukaryotes, they're highly conserved. So the same class of transposons, which I'll define in a moment, the same families, huge families, can be found in bacteria as well as in plants and humans and everything else. So they're really, really ancient and very important components of the genome. For example, the ACDS transposons, activator and dissociation, that she found in maize. There are homologs in flies. It's called hobo in flies. And there's even a defective copy in human called trap. You can see that there's a theme here to the names of transposon. Well, they're typically named after things that move. So, for example, my favorite one in Drosophila is called HMS Beagle, which is the ship that Darwin sailed around the world. There are other classes, though. So, for example, the TC1 class in C. elegans or Mariner and Hobo is a separate class. They also go all the way back to microbes, but they're distinct from the ACDS class. So these are really ancient things. And each class is defined by the gene that they encode, the transposase enzyme that allows them to move around the genome, and the homologies between those, and also between the intermediates and sequence preferences and so on that those transposases, they're sort of like restriction enzymes, can confer. Just as an example, here is AC itself. So this is the autonomous version of dissociation, DS. It was cloned in the 1980s. It was cloned molecularly, so the DNA was identified in sequence by Sue Wester and Nina Fedorov, working in the 1980s. They used the waxy gene to identify AC, and actually Joe Messing ended up sequencing the AC element. And since then, we've recognized that it really is remarkably similar to the Drosophila and human elements here, for example. And importantly, it produces the same target sites, the 8 base pair target sites in all those organisms. And that's because the transposase is like a restriction enzyme and cuts a staggered 8 base pair site. If you just do for those scientists in the audience, if you do a blast search against GenBank, you'll find this homology going not just through plants, but also through animals and microbes and so on. So she really had discovered something that was fundamental to biology and highly conserved. Since then, people have been able to actually reconstruct transposons. So, for example, the Sleeping Beauty transposon was reconstructed from a bunch of fish bought at a fish shop. And by some clever informatics, they were able to deduce the consensus sequence and get an active transposon. And when you do that sort of experiment, or for that matter, if you use the plant ACDS elements, they transpose in other organisms. So ACDS transposes just fine out of the plants. We've actually used it extensively in Arabidopsis in our gene trap program. But it also transposes in fish and in yeast. So these transposons can really move around. But importantly, sometimes in closely related species, they don't transpose. So P elements from Drosophila, a very famous transposer from Drosophila melanogaster, do not transpose in a very closely related species. And this turns out to be due to epigenetic forms of control. And I'll get back to that in a minute because, of course, Barbara discovered that too. I just want to also do a little shout out to the yeast mating type group at Cold Spring Harbor. It turns out that mating type switching, which was very famous here in the 70s and 80s, maybe early 90s, discovered originally by Ara Herskowitz, who's shown here with Barbara at a Cold Spring Harbor seminar, and Jim Hicks, Jeff Strathern, and of course the late, great Amar Clark were all working on mating type switching in yeast. And it turns out mating type switching is controlled by domesticated transposers. We now know this, they didn't really know it then. And in fact, many different yeasts have different transposers that allow them to switch their mating type. Perhaps most dramatically, our own immune systems, our own immunoglobulin rearranged antibody genes are also rearranged by domesticated transposons, a Mariner class transposers called RAG1 and RAG2. And so what she discovered is fundamental to much of what we consider in biology, and it was that discovery was made in 1979, I think, Tom Gower. And she got the Nobel Prize in 1983, I think in part because of the recognition of how widespread this really was. But it did take some, whatever it was, 35 years for people to realize that. Okay, so I did briefly mention retrotransposons. As I said, there are two classes of transposons, the ones that she discovered were these DNA transposons. But actually the most common in the human genome, and actually in plant genomes as well, are retrotransposons, and these go through an RNA intermediate. And for various reasons, I'm going to now explain what that is. Retrotransposons, again, fall into two different classes. They both use RNA intermediates. The first class are LTR retrotransposons. These were the first to be discovered in the 70s and 80s. They fall in different families. And the other rather unimaginatively described as non-LTR retrotransposons. This is like a negative space. And these turn out to be the most abundant in the human genome. So about something up to half the mammalian genome is full of these non-LTR retrotransposons, and these will become important later even in plants. Just to give you a brief one-slide history of LTR retrotransposons. They have long terminal repeats, that's why they're called LTR, shown in blue here. So the sequence, the DNA sequence is identical at the ends of these retrotransposons. In contrast, non-LTR retrotransposons, as I said, unimaginatively don't have LTRs. That's why they're called non-LTR retrotransposons. But they do have some other features like this polio tail. LTR retrotransposons are very similar to retroviruses. And if you don't know what a retrovirus is, HIV is a retrovirus. And they actually all move in much the same way. They use the same types of genes. There are obviously very important differences, including particles that allow them to form a particle within the cell, proteins that do that, and a protein that has multifunctions, including the critical reverse transcriptase, which is the way in which RNA is made into DNA to allow them to move around the genome. The way they do that is they use tRNA primers to prime that reverse transcript, that first strand DNA synthesis, and that happens at one of these LTRs. And that's why the LTRs are identical, because that DNA can now be used to prime the other end of the RNA, and that's how you end up with an LTR retrotransposon. That's greatly simplified, but it's an explanation for why they form the way they do. But tRNAs turn out to be very important in this process, as does something called PBS, the primer binding site, which is where these tRNAs bind to the retrotransposon. And if you think about it, that primer binding site is almost a very sensitive part of the sequence that has to be absolutely conserved to match that tRNA. And that's true of HIV as much as it is of these retroviruses. And the first retrotransposons, I think, if I get this wrong, but someone I'm sure will correct me, were discovered in yeast by Jeff Becker, who was with Jerry Fink. Jerry was actually a very good friend of Barbara's as well. They talked a lot about transposons. Non-LTR transposons were more recently discovered. As I said, they turn out to be the most abundant in the human genome. They're also important in plants. And they use a different, very simplified way to integrate into the genome. That's the RNA at the top, that's the DNA here. They find a site that has a stretch of A's, and they use the T's on the other strand to prime reverse transcription. So those T's, much the way as we would do in the lab, if you're making a cDNA library, you use oligo-DT. Well, non-LTR retrotransposons do the same thing, only they use that stretch of T's from within the genome, and so they end up integrating into those places. And that gives them a pattern of integration that I'm not going to get into, but it's all very, very important stuff. As I said, these are actually the most abundant non-LTR transposons in the human genome, and they're quite abundant in plants and other organisms as well. Okay, so after that brief molecular discussion, McClintock, Barbara, after her discovery of transposable elements in the 1940s, went on to look at their properties in terms of gene regulation. And she didn't actually call them transposable elements. She didn't find transposition itself at all surprising. And in fact, other major geneticists didn't either, because she was in competition with R.A. Brink and others who were also identifying the very same transposons at the same time. There's no doubt that she won that competition, but transposition was something that most geneticists at least were perfectly happy about. What was controversial about her ideas was that the idea that these transposons could control genes. And she had really good evidence that that was the case. And she used her own nomenclature to explain this. And one of the words she used was cycling, which at the time no one understood. And I hope you do after this slide. If not, I haven't done a very good job. But what she showed, demonstrated genetically, was that transposons control genes. If a transposon lands next to a gene, it can turn that gene on and off, even without jumping up and down. It can do it, it turns out, through epigenetics. So when transposons integrate regulatory regions, we now know, for example, a promoter or an intron, they can then bring the gene under their control by changing those regulatory sequences. And all transposons, it turns out, do this. And this has only been recognized much more recently, but even Drosophila P elements, for example, every transposon in maize does this, in Neurospora and in mouse, as I'll show you in a moment. All of these transposons can regulate genes. And this was what she was really excited about. In fact, she wrote an article in the American Naturalist, I think, comparing her discovery to that of Jacob and Monod, because they had discovered a repressor and an operator, and these controlled genes. And she saw her transposase as a repressor, and her transposon integrated into regulatory regions as the operator. And she made that parallel. And in fact, there's a wonderful picture of her and Jacques Monod, which is absolutely my favorite picture of Barbara, where they're laughing their heads off at the 1954 symposium, I think. Probably about this very thing. And anyway, so she really was much more interested in that than in transposition. I just want to give you one illustration of work that was done in the 1980s to sort of get to the molecular grips with this. This is actually work from Heinz Sader's lab in Germany. And this particular, we actually have this allele in the back of the room as well. This is an SPM, she called Suppressor Mutator element. You get the idea, it suppresses genes. That is integrated in an intron in this case, and this will become relevant later on as well. What she noticed was in the presence of the transposase, you get these clear colored kernels with spots. The spots are because the transposon jumps. Okay? But importantly, the background color is clear. But in the absence of the transposase, the gene comes back on again. Even though the transposon is still there, the gene comes back on. And that is why she thought that the transposase was acting like a repressor. It was repressing the gene, suppressing it in her terminology. And we had long arguments about the word suppression. Actually, I didn't. I completely agreed with her use of the word, but other people didn't. And that's why she had that view. And this is just what that air looks like. You can see this again at the back of the room. This is that exact allele of the A1 gene. And you can see that some kernels are clear with lots of little dots, and others are fully colored. And those fully colored ones are ones where the gene has come back on. So she saw these controlling elements as being fundamental to the way in which genes are regulated and to the way in which plants and animals and everything else develops, because we all knew by that time that gene regulation was key to development. For example, Ed Lewis was a good friend of hers, and she talked a lot with him about genes that we are now studying having to be like polycomb. But in fact, she was sort of wrong about that. Transposons are not responsible for the big developmental changes that have happened during ontogeny. But over evolutionary time, they have definitely had a huge impact on development. So in many ways, she was right. The important thing about cycling is that she realized that these transposons could turn on and off even without moving. So it got even more obscure here. Not only is the transposon still there at the gene, the activated transposon is also still there, but somehow it's gone off. It's turned off. And we now think of that as epigenetics. So in fact, she had some of the very earliest examples of epigenetics. She referred to epigenetic turning on and off as cycling, and changes of phase as changes of epigenetic status, and using all this nomenclature just completely confused everybody at the time. But it was fully understood by some of her contemporary major geneticists, Alexander Brink and Ed Coe, who had discovered something similarly epigenetic called paramutation, which has since become very famous. Vicky Chanda, for example, has sorted all this out. And they had huge rows about whether paramutation and transposon silencing were in fact the same thing or not. And I can tell you now that they are. In fact, they both involve RNAi, DNA methylation, and histone modification, all of that. So they were in fact the same thing. But Brink, at least, and McClintock had terrible rows. I had the honor to bring Ed Coe to see Barbara before she died, and they were still pretty prickly about dinner. So it turns out, of course, that epigenetic regulation is what Barbara had discovered. And the easiest way to think about it, and I know there are two types of audience, there's scientists here who think about the nitty-gritty of how this works, and there's other people who DNA methylation probably doesn't mean much. DNA methylation is an epigenetic mechanism. It's a way of chemically modifying the DNA without changing its sequence. And you can remove those chemical modifications just as easily as you can put them on. And so it's a reversible, it cycles on and off. So it really has the properties that you want for her cycling. And it turns out, we now know, transposons are very heavily methylated in all organisms that have DNA methylation. Now, not all organisms have it, and I'm not going to get into the nitty-gritty of that. We understand sort of why. But many do. Mammals do, for example. Plants do. Many fungi do. And DNA methylation is very important for transposon control. And this is actually, I stole this structure. The structure of the very first methyltransferase was discovered here at Cold Harvard by Saulius Klimasauskas, who was working with Rich Roberts at the time, and it's a very famous structure. And it was published actually just shortly after she died. So this is where I come into the picture. So Mila wanted me to sort of talk about how my science is related to Barbara's. I joined Cold Spring Harbor almost exactly 30 years ago in January 1989. This picture was taken, I think, the following year, I'm pretty sure, in 1990. It's actually taken in the greenhouse. Many people think it's a cornfield, but actually it is the greenhouse. What she's showing me how to do there is a technique that all those beautiful chromosome stains that I showed you before, they're at meiosis. So this is a very specific stage of germline development. And to isolate those cells, you actually have to perform surgery on the maize plant. And she would use a razor blade cut the maize plant open, peel back the leaves, and pull out the developing sporocytes, the tassel, if you like, but developing, and then pick off the myocytes and put them on a slide to stain. And it was my graduate student Ben is laughing about this because he's still trying to do it now. And anyway, I was never very good at it, but she was really amazing. Even at 90 years old, she wasn't quite 90 years old, she could still do it, and it was really, really spectacular. One of the reasons I was excited to come to culturing, however, was because I had already been working on transposon elements with Mike Freeling at the University of California in Berkeley, and I'd actually been working on cycling, maybe without realizing it, but I was. I was working with a different maize transposon. It's called Robertson's mutator, but it doesn't matter. All the transposons behave much the same way. And I discovered that there was a gene that controlled chlorophyll pigmentation, whether it's light green or dark green, called HCF46. And there was a transposable element in the promoter that, when it got methylated, turned the gene on. So that was very similar to her suppressor mutator, her A1 mutant, which we have in the room. And in a couple of papers, we were able to show, literally by cutting out those stripes with a pair of scissors and doing southern blots on the remaining DNA, for those of you who know what that is, that in fact the dark green sector had methylation at this transposable element, and the light green sector did not. And this was published first in Genes Development, but then in PNAS, and actually that paper I thought for a long time was the last paper that Barbara submitted to PNAS. It turns out I'm wrong. Rich Roberts got one in afterwards. He told me recently, and so it was actually not quite the last. But anyway, it's this ability of gene control that Barbara discovered, and the fact that DNA methylation can influence this control pretty dramatically and cycle on and off, has of course now been found in essentially all organisms that have DNA methylation. This is just a beautiful example from my good friend Emma Whitelaw in Australia, who found a very similar example in a mouse, where a retrotransposable element now is integrated into upstream of the agouti gene and conditions the coat color of the mouse. And you can see that mottled coat color is that same variegation, the same on-off cycling that you can see in maize, both in bone material and many other examples. So that was all very satisfying. But a big difference between the maize cycling transposon methylation and the mouse cycling transposon methylation was that in maize, if you take a plant like this and cross it to another plant in the next generation, that methylated form is inherited. And it's actually inherited forevermore. And paramutation is even worse. So it's very stable and does not get reset. Whereas in the mouse, if you take these mice, any one of them, and cross them, the next generation will have exactly the same range of variation as this one, pretty much. There's some influence of environment, but not much. So there's some difference between plants and mammals that in whether they get reset in the next generation or not. And actually, again, Barbara used terms just like that. She talked about erasure and resetting as to explain this type of differences in inheritance of this time. Well, we know why DNA methylation is not inherited very well in mammals. There are examples, but generally speaking, it's not. And the reason for that is that DNA methylation in mammals gets heavily reprogrammed in very early development. So in the embryo of a mouse, for example, shown here, DNA methylation levels drop precipitously in the early embryo, in the pre-implantation embryo. They then come back up again as the embryo matures, but then in the germline they go down again. So it's like you're scrubbing the genome clean twice in the embryo to remove all this epigenetics and reset the way it should be in order to start the next generation. And this is called reprogramming. It's a subject of a huge amount of research, and it's very, very important for all sorts of diseases and so on. So that's why it's not inherited particularly well in mammals. Now, I'm not saying it's not completely. There are some transposons, including the one of the agouti, that escape one but not both of these scrubbing events. So there's some leeway here, but generally speaking, the genome is reprogrammed. And it turns out that those transposons that are turned on, so I should say, obviously, most of this methylation is in transposons, the stuff that's getting reprogrammed. And so transposons come on during embryo development. That's bad news. You don't want rearrangements of the genome, chromosomal breaks, all that sort of thing to happen. And so mammals have evolved numerous ways to silence those transposons, even though they're unmethylated. Most famously in the germline, of course, are pRNAs. Greg Hannon and many others have worked on these for a long time, and we understand them very well. At this stage of development, it turns out pRNAs are not present, and instead, another form of small RNA is used instead, and Andrea Schorn discovered these. These are tRNA fragments that actually mimic, remember that tRNAs are used to prime these transposable elements, and these tRNA fragments mimic that tRNA and block that step in transposition. So even though the transposons are being transcribed, they can't transpose. And so this is a very important way of regulating them. And it targets this special region of the primary binding site, which will become relevant again in a moment. So what about plants? So I didn't realize when I was isolating those myocytes with Barbara that I was actually going to end up working on male meiosis and pollen many years later, but in fact I did. So quite thankful for that. So plants, of course, also have germlines, but they inherit epigenetic marks to some extent. And so why is that? What's different about reprogramming of the genome in plants? So plants, at least pollen, is a relatively simple system to study this sort of thing. Shown here is a pollen grain of a Arabidopsis. Those two green cells are the two sperm cells. One of them goes on to fertilize the embryo, the other fertilizes the endosperm, which you can think of like a placenta, except that it's has two parents. Those sperm cells commit their DNA to the next generation. The red nucleus is different. The red nucleus you could think of like a companion cell, a support cell, and it just helps the pollen tube grow. It does not contribute its DNA to the next generation. Okay, so a number of years ago, we set about sequencing the methylomes of those different cell types to see what happens in genome reprogramming in plants. And Joe Gallarco and Philippe Bosch were able to put together a very nice summary here, where in fact what we discovered is, this is just a diagram of germline in plants, that much of the methylation found in plants, this is called symmetric methylation, CG methylation, doesn't change in the germline. It stays exactly the same in the microspore and the sperm cells. So in fact, that might explain why epigenetic inheritance is much easier in plants, because it doesn't do this scrubbing in early development. But you do lose some of this methylation in that vegetative nucleus. And even though it doesn't contribute its DNA to the next generation, this turns on transposons in that vegetative nucleus. And they start to produce small RNAs, which I'll get to in a minute. Importantly, about half the DNA methylation in plants is actually not in this CG context. It doesn't have a G. And that really does get reprogrammed in much the same way as in mammals. So it drops precipitously in meiosis, stays very low in sperm cells, and then comes back up again after fertilization in the embryo. So this is classic reprogramming of about half the DNA methylation in plants. But at the same time, the vegetative nucleus is producing all these small RNAs. So Keith Sockin, when he was in the lab 10 years ago now, did some very nice work to show that the loss of DNA methylation in these vegetative nuclei results, first of all, you can see in that sort of expansion, almost explosion of chromatin, because it's losing its heterochromatin compared to the sperm cells. This turns on transposons, and the transposons then start making a lot of small RNA, and that small RNA, amazingly enough, ends up in the sperm cells. It moves into the sperm cells. So although the vegetative nucleus is not contributing its DNA to the next generation, it is potentially contributing small RNAs. We wanted to know how these small RNAs were generated in pollen, and Philippe found a very, very abundant microRNA in pollen called miR845. It's really abundant in pollen, not as far as we can tell anywhere else in Arabidopsis, only in pollen. And once again, it targets the primer binding site of LTR retrotransposons, exactly like those tRNA fragments do in the mammalian genome. And that's because the primer binding site can't mutate. If it mutates to try to escape the small RNA, it won't be able to prime anymore. And so it won't be able to transpose anymore. So it's a uniquely sensitive site for transposon control. And what Philippe showed was that in Arabidopsis, almost all these retrotransposons have the same tRNA, use the same tRNA, and so have related primer binding sites, and that makes them uniquely sensitive to this microbe. And as I mentioned, we'd already shown in the mouse that the primer binding site was also targeted by small RNAs. And that's this idea that actually the primer binding site of retrotransposons is almost like, Andrea sometimes likes to call it the Achilles heel of transposons, because it can't mutate, it can't change and get out of the scenario. Philippe went on to show, I'm not going to dwell too much on this, that this microRNA, because it targets transposons, generates a ton of secondary small RNAs specifically from transposons, not from genes. You don't want to silence genes, just from transposons, and that's the selectivity that allows transposons to make all this small RNA. And interestingly, it's found in some, on the left, varieties of Arabidopsis. That black line shows you the secondary small RNA. But some, interestingly, don't have it, don't have this microRNA, which is weird because it's highly conserved and produces all these sinuses, transposons. So how come some Arabidopsis don't know it? So this is where I get back into history again. So I want to introduce you to Albert Blakesley. He was the director of Cold Spring Harbor, the Station for Experimental Evolution, back in the 1920s. And he worked on evolution, and he worked in plants. And he was growing his Jimson weed here in the parking lot. It wasn't a parking lot in those days. And he found one out of many, many thousands of plants that looked a little different. It had slightly larger fruits. It just looked a little different. But importantly, when he selfed it, it produced normal amounts of seeds. But when he crossed it back to the progenitor, the field of Jimson weed that it came from, all the seeds died. And so this is the definition of a new species. He was very excited about this. He published a paper in Scientific American saying how he discovered a new species, this was experimental evolution, and so on and so on. Well, a couple of years later, he realized that actually it wasn't the new species. It was a tetraploid. It had doubled its genome. It had doubled the number of chromosomes. And because of that, when it crossed back to the diploid, the progenitor, it makes triploids. So we all have two genomes, one from mother, one from father, so we're diploid. If you double that with a tetraploid, you have 4 genomes. And if you cross a tetraploid to a diploid, you have a triploid, that's 3 genomes. And he called this, well actually, it was subsequently called the triploid block. This is the reason why seedless watermelons have no seeds, all that sort of thing. It's conserved among most flowering plants, and he went on to show that 20 different species did the same thing, and that's what this paper was. You're wondering why I'm telling you this. This was published almost exactly 100 years ago. Well, it turns out, that, oh yes, had to show you this. Albert Blakesley was the director who first hired Barbara as a temporary visiting scientist for a year. This is the acceptance letter from Barbara written to Blakesley. She ended up staying, and it was Demarek who actually made it the permanent offer. But it was Blakesley who was the director at the time in 1941. Okay. So it turns out that the triploid block works really well in Arabidopsis. We can reproduce it extremely well. For example, in this Colombian landrace Arabidopsis. But interestingly, the Landsberg that did not have the microRNA doesn't do the triploid block. So maybe the microRNA is responsible for the triploid block. And in fact, that turns out to be the case. So we collaborated with Claudia Köhler at the Uppsala, actually the Linnaean Institute at the Uppsala University in Stockholm, in Uppsala, who's the expert on the triploid block. And sure enough, we were able to show that if we mutated, if we made mutants in the microRNA that no longer have it, now the triploid block is removed. And so the seeds are perfectly normal, even though they're still triploid. We were extremely interested to find out why this was. It turns out that the small RNAs that are triggered by these RNAs target imprinted genes. Those imprinted genes control the balance between genomes and the dosage between genomes. And if you think about it, what better way to count chromosomes than to count transposons? Because every chromosome has a ton of transposons on it, and if you double that, you end up with a big, significant difference in the number. And so small RNAs and target transposons are an excellent way to estimate the number of chromosomes in your genome. So this sort of makes sense. And this was much along with Claudia a few years ago. Earlier last year, 1 year. All right, so I just want to finish on one last example of the importance of transposons in plant breeding. So what I've been telling you about is reprogramming. So when plants produce seed, or when mammals reproduce, they go through meiosis, and fertilization, and everything gets reprogrammed and reset for the next generation. But what happens if you don't do that? So many plants can clonally propagate. Actually, a lot of animals can too. They can propagate clonally, and actually one of the most dramatic examples is duckweed, some species of which probably only propagate clonally. When they do that, they don't go through meiosis, they don't go through fertilization, and so what happens to their genomes? What happens to their transposons? Does something change with respect to gene control? Well, there's a classic example of this, which I've had the pleasure of working on for the last 20 years or something, which is the cloning of oil palm trees. They're not really trees, they're oil palms. And this was a technique that was developed in Malaysia in the 1970s, where you can select an oil palm that is very high yielding, doing extremely well when it's mature, so you can measure the yield. Take a tiny piece of the youngest leaf, put it into tissue culture, and regenerate up to 50,000 genetically identical copies of that high-yielding palm. And this was supposed to be the savior of the oil palm industry, because breeding oil palms takes a very long time, a 10-year cycle. And so this was supposed to save their industry. However, it turns out that not all clones are identical. This slide was actually made for me by Joe Clark, who was a huge movie buff. It's well known in plants that if you do this sort of micropropagation, you get a lot of variation that's not genetic. They're genetically identical, but something else is happening. Guess what? In oil palm, a very frequent, highly variable, but very frequent abnormality that arises in these oil palm plantations is called mantled. This is what a normal fruit bunch looks like. You can think of the fruit, it's called a drupe, it looks a lot like an olive. In the mantled case, you get shriveled fruits. It's actually caused by a homeotic mutation that converts the stamens, the male organs, into female organs, and as a result, it's not fertile, it doesn't set seed, and so on. This is a huge problem for the industry. They basically stopped cloning. The only people who can afford to take the risk of cloning are the big seed companies and not the smallholders. And so it greatly reduced the impact of this potentially important technology. We set about trying to find methylation differences in the oil palm genome that might explain this mantled abnormality. And we used what I like to think of as trios. So you have a mother palm, which is high yielding and has been cloned. And then you have two clones, one of which is normal, the other of which is mantled. And those three palms are genetically identical, right? They have the same DNA sequence. But epigenetically, they may not be. And we used a technology that we actually developed here at Cold a very old-fashioned microarray-based, very cheap microarray-based way to do methylation, which is invented by Sackman and others in the lab years ago, and examined hundreds and hundreds of these trios. And it turns out there are thousands of differences in DNA methylation, most of which are randomly distributed between them all, but there's just one site in the genome which is always unmethylated in mantled and always methylated in normal. And it turns out to be a transposon. It turns out to be a transposon in an intron of a gene, just like Barbara's SBM is in an intron of that gene sitting at the back of the room. It's actually a line, a non-LTR retrotransposon, and it's from a family of transposons. I mentioned that transposons come in families, which are highly conserved. Family of transposons that are already being discovered and named in rice, and it had been named Karma. So it was named Karma because it had an intergenerational effect on transposition in rice from tissue culture. And it's the same type of transposon in oil palm that's causing the mantle gland mounting. And interestingly, what's happened is it's lost DNA methylation, but it's only lost DNA methylation in mantle gland here, in the non-symmetric, non-CG, the type that gets reprogrammed in pollen. In other words, because it hasn't gone through reprogramming, it's lost its DNA methylation, it's lost it permanently. This actually interestingly results in a splicing difference, a very dramatic splicing difference, only in mantled palms. That splicing difference means that the gene becomes non-functional. It's probably the same mechanism in Barbara's A1N1. Sitting in the back of the window. It hasn't actually been shown. Maybe we should grind up some of that corn. No. And it's actually a very interesting observation that this would cause this. But important, and of course this gave us the opportunity to name the epi-alleles, right? We could call the methylated allele good karma and the unmethylated allele bad karma. And in fact, interestingly, we see exactly the same shift in small RNAs that you see in pollen during reprogramming. So from 24s to 21s, I didn't go into that difference, but it really looks very, very comparable to what we see in Arabidopsis. And finally, and most importantly, we were able to develop a simple DNA methylation test that can distinguish between good karma and bad karma before the plants go into the plantation. And that's important because once they're there, it's really hard to, you can remove them, but you've lost the use of that land. And this is highly, highly valuable and important land. It also led to my favorite journal cover ever, which was Weed Out Bad Karma. So weed out the bad karma. And I should say that this work was done with the Malaysian Palm Oil Board and with a company of which I'm a co-founder, Orion Genomics, of course, being a spin-off company. We like to think that this is important because oil palm, of course, is a hugely controversial crop. It's grown in the middle of the rainforest, and part of the problem with oil palm cultivation in the rainforest is that if you have low yield for whatever reason, bad karma, for example, the temptation for smallholders to burn down the neighboring rainforest accidentally and then plant more oil palms there is enormous. And so having these tests, we like to think, will make a difference to restricting oil palm encroachment on the rainforest. This is a picture taken by Elena, who was the first author on that paper. She got into a helicopter and took a photograph of Borneo, and on the left is the oldest rainforest on Earth, supposedly, paleontologists tell us. And on the right is an oil palm plantation. And you can see the change in biodiversity. It's pretty spectacular. So I just want to end with this this last and perhaps my favorite photograph of Barbara. It's not a particularly good reproduction. She's in a cornfield in Mexico with two of the most famous Mexican maize breeders of all time. She's actually researching a book at the time that she subsequently published on the origins of maize, and she was able to use that staining technique again to trace heterochromatin patterns, so chromosomal banding patterns, into the Guatemalan highlands, where she thought that maize must have first been domesticated. And she's just got this fabulous laugh, which I think sort of expresses how she must feel about transposons now. Interestingly, and this is Dave figured this out, the young boy standing next to her, I take no credit for this, it was all Dave, is actually the son of Norman Borlaug. Norman Borlaug was the father of the Green Revolution and won the Nobel Prize for the Peace Prize later on, and that's his son. So it's pretty cool. All right, I'm quickly though going to acknowledge members of my lab. You probably can't even read that here. Former members of my lab, Philippe and Joe, did all the work on pollen epigenetics with Jean-Sébastien and Rowan, and they have since gone on to better things. Andrea, of course, with some help from Michael, did the work with tRNA fragments and retrotransposons in the mouse. And I should probably thank a whole bunch of people, but these are some of the people in Malaysia who were very important in the oil palm work. But I'll leave you with that picture.